finally, the actual genetics that you've all been waiting for. So again, this is familiar for many of you, but this is revision, and for some of you it will be new. So the genetics of PWS can seem complicated, and in some senses it is, but it's very, very interesting. And it's based upon having an understanding of a basic abnormal situation on chromosome 15 and different ways in which it can be abnormal. So this white square here shows you two normal situations. For the vast majority of genes across the human genome, you get expression of genes from both a paternal and maternal chromosome. And those are non-imprinted genes. So this could be any chromosome. And the yellow ring here would be the genetic sequence for a non-imprinted gene that is equally expressed from the male and female, the paternal and maternal copies. At only around 150 loci across the genome, so not very many places, imprinting has occurred. Okay, putting aside the X chromosome, which is different, so autosomal genes, there's only a very small number that are imprinted. And what does imprinting mean? It's a process by which early on, very early on after conception, one or other of the copies of a gene, set of genes, is switched off. So in this example, in this normal example, the maternal copy here has got a red cross on it, meaning that methylation or other modifications of the chromosome led to switching off of expression of that gene. Expression meaning transcription of that DNA sequence to make RNA and then translation to make protein. For imprinted genes, it's extremely important that the DNA sequence of both the paternal and the maternal copies are normal, because if there was an abnormality here of the paternal copy and that gene wasn't expressed, then the individual would have no expression at all of that relevant gene, and that protein would be missing from their cells, and whatever job that protein is doing wouldn't be being done by that protein. Okay, so that's an explanation of why imprinting is so important. So what happens in PWS? It's a disorder of an imprinted locus, chromosome 15. And the disorder is that in a number of different ways, the paternal copy of this region of chromosome 15 is disrupted, so that because the maternal copy is already imprinted and switched off, there is no expression of the genes at that locus. It can happen a number of different ways, but that's all you need to know. You've got a loss of the paternal copy of the imprinted locus. How can that happen? It can happen in three different ways. First of all, that region can be just deleted, can be absent. So here, this blue paternal chromosome is shorter because the relevant section up here has been deleted. That's one way. The second way is called uniparental disomy, UPD. And in this situation, rather than having a paternal copy of the chromosome and a maternal copy, there are two maternal copies. There are a couple of different ways that can happen during meiosis with an early segregation of the chromosomes, or in fact, after conception in a post-zygotic change, an individual will, in either all or majority of the cells of the body, has only the maternal copies of that chromosome. There is no paternal copy. Both copies are imprinted, and there's no expression of genes at the imprinted locus, UPD. The third way that you can have loss of the paternal copy of 15q13 is by a problem with the imprinting process itself, whereby the paternal copies that should be switched on are switched off. And that may be because the imprinting center that I'll show you has been deleted, or we have a DNA sequence difference with it, or perhaps because a distant genetic problem, some other place in the genome, is causing abnormal control of imprinting. This is a much rarer cause, but and still a subject of research. But that's, in broad principle, the third way in which you can get absence of expression of the paternal genes. Here is a cartoon of the PWS region. For those of you who don't know it, it's quite complicated. Here you can see the two typical deletions, typical deletion 1, typical deletion 2, one going from breakpoints 1 to 3, the other from breakpoints 2 to 3. There's no good evidence that I'm aware of any genotype-phenotype correlation dependent on deletion size. And there's lots of research ongoing about which of the genes in the deleted region and in the PWS region are critical. It's probably not just a single gene like MAGEL2, for example. It's a combination of lack of expression of both a number of genes in the PWS region and the non-coding RNAs that are probably contributing to phenotype. How common are each of the different genetic causes of PWS? Well, based upon the global PWS registry, which published in 2019, around half of PWS is caused by the deletion, around a third by UPD, about 3% by an imprinting defect, and then there are rarer causes like Shafiang, which is a single gene deletion, I think, and translocations. And in the registry, 10% of cases didn't know what genetic type of PWS the patient had. Does it matter? First of all, now there's been an enormous research drive to compare, for example, deletion and UPD cases and see whether the frequency or severity of the different clinical characteristics are influenced by the type of genetics, and I'm not going to cover any of that today. Does it matter from a clinical genetics and counselling perspective? There it does. So for both deletion and UPD, after a couple have had one child with PWS, in general terms, the chance of it happening again is low in either of those circumstances, but not always. So you may have a predisposing carrier type, like a translocation, which increases the chance of UPD. And it's important to pick that up to give families an accurate prognosis in terms of the risk for future children. And similarly with imprinting defects, the type of change in the imprinting center might change whether we say that the risk of future children having PWS is low or high, and whether prenatal testing is a sensible option for families. So genetic testing, given this complexity, where would you begin? How would you know what tests to order to diagnose PWS? Well, the specific subtypes are complicated, but the unifying reason for PWS, that loss of expression of the paternal copy, is easy to test for with a single test. And so that's the step one, the testing for the paternal expression of the 15q13 region. There are a number of different tests that are used to detect lack of paternal expression. And in the UK, we're mainly using methylation-specific PCR of one of the specific exons of one gene in the region. There are other technical tests that one can do, but that's the one that we mainly use. And it's 99% diagnostic, i.e. if a person has PWS, in 9% of cases, that test will be positive or will show lack of paternal expression, and that PWS is the only cause of that test result that there can be. Why is it not 100% diagnostic? Because in theory, you can have, or not in theory, but in very rare circumstances, you can have mosaicism whereby the initial test might be normal from a blood sample, and you might need to do a buccal swab to detect whether PWS is present in some cell lines and not others in the human body. So if you are absolutely certain that somebody clinically had PWS, but their methylation analysis came back normal, you might want to look for mosaicism. If step one shows abnormal paternal expression, you've diagnosed PWS. You can stop there if you don't have access to other tests, but you would go on to determine whether somebody had a deletion or UPD or an imprinting defect. First of all, you would look for a deletion because that's the commonest situation in 50% of cases. And depending upon where you are in your local lab practice, you might have done that in the old days by a FISH test, but now you would do it by a chromosome microarray. And in fact, for many people now, the chromosome microarray may have been the first test that they had, and the first result may be that the deletion is detected. And you would follow that up with a carrier type to be sure that there wasn't a paternal translocation predisposing to deletion. Step 3, you'd only need to go on to if you didn't detect deletion. So step 3 is testing for UPD, and you would do that by needing samples from both a mother and a father. It's only possible if both parents are available for testing. And you would do that either by looking for microsatellite markers on the paternal and maternal chromosomes and testing that the baby carries both the maternal and paternal copies of chromosome 15, or you might do that on something that's called a SNP microarray. Lastly, you would only go on to look for an imprinting defect if step 3 was also normal. So you could do that via targeted deletion testing at a specialist lab. We have one centre in the UK that would do that testing, and that would also sequence the imprinting centre if no deletion was detected. If that was still normal, so if you had abnormal methylation, and so you would diagnose PWS, but steps 2, 3, and 4 were all normal, then you'd be looking for a very interesting case where you might be looking for an epigenetic mutation somewhere else in the genome, and that would be research testing. So that is your full diagnostic testing pathway at present for PWS. That's where I'm going to stop. I've raced through that, I know, probably quite quickly, but the testing materials, my slides will be available from Shelley later on today. And the IPWSO website describes this testing pathway in much clearer detail than I have done. So I've provided you with a set of information so that we all have that shared knowledge about the lifespan clinical characteristics of PWS. I've explained the genetic basis for PWS, and I've raced through the steps of diagnostic genetic testing. So I hope that you understand why step 1 is the most important, and steps 2, 3, and 4 are important, but they're important for genetic counselling about recurrence risk. And I know that those tests will not be available to all patients. And I hope that you now can recognise a person who may have PWS at any point in the lifespan and think about how to initiate diagnostic testing pathway for them. Thank you very much.
Thank you, Kate. That was a great summary of clinical presentation and genetics.